Good day, Dr. Huda. Our group project title is Lung Cancer Histopathology Teaching System. Let's start our presentation today by introducing what is lung cancer. Lung cancer is the most common and deadly form of cancer. It accounts for 1.6 million new cancer cases and approximately 1.38 million people die from lung cancer annually. In fact, 95% of lung cancer patients die within 5 years of diagnosis. Lung cancer cells can progress rapidly, even in the early stage. As such, early diagnosis is the key to treatment. However, most lung cancer is diagnosed too late for curative treatment to be possible. In over half of people with lung cancer, the disease has already spread at the time of diagnosis. Early diagnosis is difficult because many of the common symptoms of the lung cancer are similar to those of smokers' lung. There are two problem statements in our project. First is the distinction between the types and the subtypes of lung cancer is a difficult task and requires extensive experience and knowledge. This is due to the complexity and difficulties in recognizing the features and characters of the cell under microscope. Second, lung cancer is divided into two major types, and the two major types are further divided into many different subtypes. Therefore, it is a deterrent for a medical student or new pathologist to make a right diagnosis on the biopsies of them. So, our project will focus on the histopathology fields of lung cancer and develop an expert system aims to teach and provide information to the non-expert regarding the classification in histologic image of lung cancer. Our system is called Lung Cancer Histopathology Teaching System. This table is published by WHO. According to the table, there are many subtypes of lung cancer cells. In general, the goal of our project is to identify the thought process of pathologists in diagnosing lung cancer using histology image and to develop a teaching expert system known as Lung Cancer Histopathology Teaching System to assist the non-experts in their learning process. There are three specific objectives. First, targeting technology as a potential tool for teaching the non-expert on histologic classification of lung cancer with the use of histologic image. Second, to observe and analyze the current practice in histopathology diagnosis process and indicate the characteristic and pattern of tissue by referring to the tissue observed under the microscope. Last one is to convert the human analysis and thought process into structure model used to develop the knowledge base of teaching expert system. This figure shows the architecture of our lung cancer histopathology teaching system. This architecture includes all basic components of an expert system, which include knowledge base, inference engine, explanation facility, and the user interface. Our system contains two modules, which are the teaching modules and the exercise module. The user of the expert system is introduced to the different histology types of lung cancer and morphologic features of each type of lung cancer seen on the biopsy in the teaching module. When users complete the teaching module, they can proceed to the exercise module, where users have to make diagnosis based on the histology image provided to them. Users have to input the features found on the tissues and choose the type of lung cancer. According to information provided by the user, the inference engine will match the features and answer input by user with the rules. Once certain rule is matched, the rule is fired, and according to the rules stored in the knowledge base, and diagnosis result is presented to the user to justify whether the user made a correct diagnosis. The explanation facility provides definition of specific medical terms, explanation on histology image, as well as relationship between features on histology image and diagnosis result. Knowledge acquisition part has been done by interviewing pathologists from UKM Medical Center to generate rules required in developing the knowledge base of our expert system. This table shows the characteristics of our expert system. Our expert system domain is lung cancer histopathology teaching system. Our knowledge resource is from textbook, research publication, expertise. Our knowledge acquisition technique is decision table and decision tree. Our knowledge representation technique is use rules, and the inference engine is forward chaining. The explanation facility is to define the definition of specific medical term, explanation on histology image, relationship between features of histology image and diagnosis result. Our expert system development method is by prototype. Development through is by Eclipse and Jest. The user interface is using Eclipse, and our main objective is to assist the non-expert in their learning process on histologic classification of lung cancer.
I'm going to present on the knowledge acquisition methods of lung cancer histopathology teaching system. To develop the knowledge base, there are 4 stages. First, we interview the domain experts, a pathologist from HUCAM, Dr. Rina, to capture her thought process when diagnosing lung cancer. Before the interview, we have prepared ourselves by reading journals and medical publications from WHO to get some knowledge on lung cancer. Secondly, after the interview, we built a decision tree based on the knowledge captured from the interview, and we also included some extra knowledge we read from journals and medical publications. Then we met Dr. Rina for a second time to verify the decision tree. Finally, with the verified knowledge, we come up with a set of if-then rules to be implemented in our system. From what we understand, the main task of histopathologists is to classify the type of lung cancer based on the microscopic images of the lung biopsy. Lung cancer are divided into 2 major types, non-small cell lung cancer and small cell lung cancer. Classification into these 2 types is crucial in the treatment plans of patients. Each type of this major type has various subtypes that are used for the assessment. The complete classifications of lung cancer histology have been issued previously by my friend Pei. However, due to the complexity and large amounts of histologic subtypes of lung cancer, experts suggested us to focus on the 4 common subtypes in Malaysia, which are squamous cell carcinoma, large cell carcinoma, and adenocarcinoma for non-small cell lung cancer, and small cell carcinoma for small cell lung cancer. We have developed a decision tree on how to classify the type and subtypes of these 4 common types of lung cancer. The classification of lung cancer depends on the tissue patterns of the biopsy. I'm going to show you the decision tree that we have developed. So this is the decision tree that we have developed. The 2 major types of lung cancer, non-small cell lung cancer and small cell lung cancer. So the features of non-small cell lung cancer are abundant cytoplasm. In contrast, small cell lung cancer has scant cytoplasm. Secondly, non-small cell lung cancer has pneumorphic nuclear coarse chromatin pattern. However, small cell lung cancer has small hyperchromatic nuclear with fine chromatin pattern. And non-small cell lung cancer, their nuclei is often prominent. In contrast, small cell lung cancer, their nuclei is indistinct. For non-small cell lung cancer, it has glandular or squamous architecture, whereas for small cell lung cancer, it has diffuse sheets of cells architecture. And the last features of non-small cell lung cancer is the presence of mucin in adenocarcinomas. However, in small cell lung cancer, mucin is absent. Squamous cell carcinoma is the first most common type of lung cancer found in Malaysia. It has a predilection in male and it carries a worse prognosis as compared to adenocarcinoma. It forms keratin pearls. As you can see from these images, this is a keratin pearls and another keratin pearls. The morphologic features suggest that squamous diffusion include intercellular bridging. This one is one intercellular bridging, intercellular bridging. These are the intercellular bridging. As for the second subtypes of non-small cell lung cancer, adenocarcinoma. Adenocarcinoma is the second most common type of lung cancer in Asia, and in contrast with squamous cell carcinoma, adenocarcinoma has a predilection in female and carries a better prognosis with 5-year disease-free survival. It usually occurs at the periphery of the lung and is a common type for non-smoker. It grows slowly and forms smaller masses than other subtypes, but tends to metastasis widely at an early age. Adenocarcinoma tumors form irregular-shaped glands. As you can see from this histology image, these are all the glands in irregular shape. Diagnosis of large cell carcinoma is further confirmed by performing immunohistochemical staining for TTF1. This is the positive immunohistochemical staining for TTF1 to double confirm that the biopsy is an adenocarcinoma type. As for the third subtype of non-small cell lung cancer, large cell carcinoma, this is the third most common type of lung cancer. These tumors can occur at any parts of the lungs. Large cell carcinoma is a diagnosis of exclusion, where the presence of small cell carcinoma and squamous cell or granular differentiation needs to be excluded by light microscopy. However, large cell carcinomas probably presents squamous cells or adenocarcinomas that are so undifferentiated that they can no longer be recognized by light microscopy. When this happens, pathologists will classify them into small cell lung cancer or non-small cell lung cancer. Histologically, these tumors usually consist of sheets and nests of large polygonal cells with vesicular nuclei. Okay, as you can see from here, this is sitting at the periphery of the mass of the tumor cells, and rosettes can be seen. Okay, these are the rosettes. Okay. The diagnosis of large cell carcinoma is further confirmed by performing immunostains of NCAM50, CD56. This is a histologic image show that a positive immunostaining of NCAM CD56 in the large cell carcinoma. As for the subtype of small cell lung cancer, small cell carcinoma, the features of small cell carcinoma are their histologic image usually show pale grey in color. As you can see from here, is in pale grey as compared to the other subtypes. Okay, tumor cells are usually densely packed, small with dense cytoplasm, as you can see from these images. They are finely granular nuclear chromatin and absence of nuclei, and metastases are frequent. Also, it features is they has like fusiform shape is prominent feature. So these are all fusiform or spindle shape. Okay, longer in shape. Okay. Also, they show more cytoplasm. Okay. These are all cytoplasm. Okay. The last features of small cell carcinoma is they have extensive necrosis. You see, these are necrosis. A decision table is derived from this decision tree, and a set of its values is generated. So this is the decision table. Show that it features 1, 2, 3, and 4. That means it's a squamous cell carcinoma types of lung cancer. And features 1 are keratin curls, feature 2 keratinization, feature 3 intracellular bridges. This is the way how we read the decision table. Then from features 5 to 11, it shows large cell carcinoma and so on and so forth. Based on the decision table that I've just showed, we have developed 4 if-then rules. Rule 1, if condition F1 or F2 or F3 or F4, then the result is going to be a squamous cell carcinoma types of lung cancer. And so do for the rules 2, rules 3, and rules 4. project, we have faced some problem. The first problem we face in this project is difficulties in meeting the domain experts. Due to the packed schedules of experts in HUKM, the interview sessions was postponed for several times until the 14th week of the semester. And the second problem we have faced is we are lack of experience and skills in interview. Due to this problem, we were only able to capture some shallow knowledge of the experts in the first interview. And the third problem that we face in conducting this project is difficulties in understanding the materials and information obtained from the internet. The journals and the medical publications mostly are written in medical terms, but we are not from medical background. That's why we have spent a lot of time and effort in understanding the knowledge regarding lung cancer as well as preparing ourselves for the interview. During this conclusion, we hope that with this expert system, lung cancer histopathology teaching system, medical students are able to learn the classification techniques of lung cancer histologic typing in an easy and efficient way. Thank you.